Great. My name is Deborah Fuller. I'm a professor in the Department of Microbiology at the University of Washington School of Medicine. I'd like to thank you all for coming. We're very pleased to announce some promising results with a new kind of RNA vaccine that was designed by Dr. Jesse Erasmus here, a postdoctoral scholar in my lab. Now, my lab has been working on nucleic vaccines, including RNA and DNA vaccines, for over 25 years. And even at the earliest stages of their discovery in the early 1990s, we recognized that DNA and RNA vaccines offered considerable advantages to protect from future pandemics, primarily because they can be rapidly designed and tested using only the genetic sequence of the virus. So this promise, as you all know, was borne out in real time for us this year as we witnessed the very first vaccine to enter into clinical trials right here in Seattle was an RNA vaccine. So in January, when the sequence for SARS-CoV-2, the virus that causes COVID-19, was first published, Jesse quickly got to work and designed an RNA vaccine for COVID-19 and was testing it in animals within 2 weeks. Now, this RNA vaccine is different from the type that are currently in clinical trials and was designed to maximize its potential to meet what we believe are going to be key requirements for an effective pandemic vaccine. Jesse will explain in more detail in a minute how our vaccine differs from these others, but let me first summarize the key findings from our paper. First, this study was performed in mice as well as preclinical non-human primate model that more closely mirrors vaccine responses in humans. As I mentioned, we believe an effective pandemic vaccine will need to meet key requirements. First, we think it will need to induce immunity in the population quickly, ideally after 1 dose or 2 doses spaced close together. In our paper, we showed our vaccine was able to quickly induce antibody responses within 28 days after a single dose, and unlike what's been seen in convalescent humans, where there are reports of rapid decline in antibody, our responses induced by our vaccine continue to go up over time and are still sustained at levels that are comparable to titers that have been shown to protect animals from infection with SARS-CoV-2. We are still continuing to monitor these animals to see just how durable these antibody responses are. Second, we think an effective pandemic vaccine should also induce T-cell responses that can help sustain the antibody and can also contribute to protection by clearing the virus. In the event antibody doesn't completely block an infection, T-cells can find infected cells and clear them. So we show in our paper that we were able to induce T-cell responses in all the animals, and importantly, they were the right type of T-cell response. Viruses like to hijack our immune systems, and if they can, and there are certain types of T-cell responses, notably called type 2 T-cells, that can aggravate an infection. That's not what we want. Instead, our vaccine directed the induction of a type 1 T-cell response. These are the good type of T-cells that can help with protection and clear the infection. Third, an effective pandemic vaccine would need to be effective in all demographics, including the elderly. As you know, elderly don't generally respond very well to vaccines. So what we show in this paper is that after only 1 dose, our vaccine was able to induce comparable responses in old mice as they were in the young mice, in all but the very oldest animals. And in these animals, we were able to bring up those responses to comparable levels as in the young animals by just administering 1 more dose. Finally, even if a pandemic vaccine meets all these requirements for immunogenicity and efficacy, it will still need to be scaled up very rapidly. As I mentioned, RNA can be scaled up very quickly, but all RNA vaccines also need a vehicle or formulation to get that RNA into our cells. This is currently accomplished with RNA vaccines currently in clinical trials by formulating the RNA with a nanoparticle technology. But getting that RNA into these particles, it's a complex process and not trivial, and could potentially pose a bottleneck in scaling up RNA vaccines to billions of doses that will be needed to stop the pandemic. In our paper, we sought to address this potential bottleneck by developing a new, more scalable formulation that Jesse is going to provide more information on. So in summary, our vaccine was able to meet key requirements for an effective pandemic vaccine. It was able to induce immunity quickly, it was able to induce T-cell responses that may contribute to protection from disease, it was able to induce strong responses in aged animals, and this was all achieved using a novel RNA vaccine design and formulation that, if human trials are similarly successful with this vaccine, should be able to be scaled up and distributed rapidly.
So most RNA vaccines are formulated by encapsulation within a lipid nanoparticle. And this results in some complexities in manufacturing because you have to manufacture your RNA and then co-manufacture with the lipid nanoparticle in order to get the RNA inside these lipid vesicle-like particles. So what we did to counter this complexity is we are collaborating with a local company in Seattle called HDT Bio. They developed a formulation called LION. This is an acronym that stands for Lipid Inorganic Nanoparticle. And what it is an emulsion, very similar to some adjuvants that have been developed like MF59. It's an oil droplet in a water emulsion. It's emulsified in an aqueous environment, a water environment. So these little lipid droplets with a surface charge that gives them a positive charge that allows RNA to actually interact with these nanoparticles on the surface. So we don't have to put RNA inside of a lipid nanoparticle, rather we can put the RNA on the surface of these nanoparticles. And this provides a major advantage in manufacturing because you can manufacture the formulation first as its own drug product and then manufacture your RNA as a separate drug product. And these two are released for clinical development independently. So you can scale up manufacturing of the formulation and scale up manufacturing of the RNA and stockpile each of these things independently. And then you do a simple one-to-one bedside mix prior to vaccination. And this was the technology that HDT had developed, and so we worked with them to get this preclinical data package that was published in the STM paper. And I think that basically covers the major advantages of the system. The formulation itself does also appear to have some adjuvant-like properties, but there's a lot more work that needs to be done to characterize that.
Let me get you to just tell us in colloquial language something we've already touched on. But a lot of vaccines are out there. What makes this one an especially good vaccine, especially promising vaccine?
Yeah, so one of the things that we have been we worked with this particular vaccine that makes it different from other ones is that it's a replicating RNA vaccine. So most of the RNA vaccines that you've seen currently in clinical trials, it's a single copy of RNA that goes into our cells and then it produces the vaccine. What this vaccine does is once it gets in the cell, everything's the same, but then it starts replicating itself, producing more copies of itself. That will produce more vaccine protein and potentially make it more immunogenic. So what we saw in our paper was that we were able to induce very strong immune responses after a single dose, which would be consistent with a vaccine that's producing much more vaccine antigen. So we think this is going to be an advantage in terms of being able to induce immunity much more quickly in the population. And then, as Jesse had mentioned, we formulated it with a novel formulation, this LION formulation, that may be much more scalable than current formulations being used with other RNA vaccines.
When you say much more scalable, just put that into terms that everybody would understand, because it's not enough just to have a vaccine, you gotta have a lot.
Yeah, you're going to need billions of doses. So I don't know, Jesse, if you can compare the particle that everybody else is using and then the new LION particle that, in terms of number of doses that can be produced.
It's kind of hard at this stage to comment on that because we don't have, we've never really stressed each of these systems in such a situation as we are in right now. And so we haven't really seen the capabilities. Theoretically, all of the components for the LION formulation are readily available and easily sourced. Some components in LNPs, such as cholesterol, are actually in short supply. And so there's potential for bottlenecks in the production process for LNPs. And then the fact that both of them have to be manufactured together requires that the final product be formulated and stored in a specific manner. So storage is going to be a huge issue as well when it comes to stockpiling and distributing these vaccines. But honestly, I can't really comment on how much of each we can make. That's something to be determined.
But it sounds like the vaccine that you have developed sort of gets around a lot of the production bottlenecks. Just explain why that's so important.
Yeah, it's extremely important to be able to get around these production bottlenecks because the demand for a vaccine is going to be huge. Honestly, there's going to be enough room for multiple vaccine candidates in the market just due to the sheer fact that there's a lot of people that need to be vaccinated. Not only a lot of people, but lots of different target demographics. There's going to be the elderly, there's going to be young, there's going to be immunocompromised, there's going to be a lot of different people that are going to require different types of vaccines. And being able to manufacture vaccines at scale that can target all of these diverse target populations is going to be key.
Do we have any questions? Are you sharing all this information so that you go to piece and then competes? The question was, are we sharing the information through our research with others?
Yeah, I did.
Yeah. Obviously, with the publication of our paper today in Science Translational Medicine, all the results that I talked about today have been published and are being shared, with other researchers. We're also collaborating with a number of researchers at the University of Washington who are going to be studying even more closely how this vaccine works. What other types of immune responses does it induce? And then, of course, collaborating with the company HDT BioCore, which is going to advance this vaccine to phase 1 human clinical trials.
How far away from human trials are you, and how long does the immunity last?
Yeah. So.
Questions, I'm going to repeat for
Yeah.
people online. The questions are, how far are we away from actually going into clinical trial, and then how long does the immunity last?
Okay, I'm going to allow Jesse to answer the question about how close we are to clinical trials because he's helping the biotech company here, and then I'll respond to the second question.
Okay, so with regard to timing of the clinical development, what I can say is that HDT Bio, which is locally here in Seattle, they've already performed the GLP toxicology studies that are required to file an IND with the FDA, and they've already had a pre-IND discussion with the FDA. And their plan is to start the phase 1 clinical trial in late summer.
Yes, and with regard to how durable the immunity is, we don't know yet. That's the short answer. To date, we've seen promising results indicating that the antibody responses are not declining like you see typically or have been reported in convalescent patients. Instead, the antibody appeared to go up and continue to be sustained. Although we haven't gotten very far along, we still are monitoring these animals. One thing that we're doing different than other groups have done is that non-human primates are a model that we can challenge with SARS-CoV-2 to see if they're protected. And one thing we didn't do in our studies is we have not yet challenged these animals. Instead, what we're doing is we're waiting until these animals have been sitting around for about 4 to 6 months, and then we're going to challenge them because most studies actually challenge their animals at their peak antibody response. But in reality, people who get vaccinated aren't necessarily going to be exposed at that time. They need to have protective immunity over a longer period of time. So we're going to wait a little bit and actually see if our antibody is not only durable, but is it protective over a longer haul.
We talked pretty clinically, but are you excited? What's exciting?
Yeah, no, we're very excited about the results, and in terms of seeing our vaccine enter into the pipeline. As Jesse mentioned, we believe it's not going to be really one vaccine that's going to knock down this pandemic. I mean, we're used to vaccines being rolled out slowly over a matter of years and getting our immunization started when we're children. For a pandemic, you need something really quickly. You need billions of doses. You need to be distributed worldwide, and I don't think one vaccine is going to accomplish that. I'm hopeful, based on some of the results I've seen, that not only our vaccine but other vaccines will be out there able to be distributed worldwide to stop the pandemic.
One question we have, and if we could repeat it, because I don't have time. How is this paper reviewed from a peer review standpoint process? Can you explain that and ask the question?
Right, yeah. So our paper today, which was published in Science Translational Medicine, has undergone peer review. And what that means is that our manuscript, which we initially posted on bioarchives, initially wasn't peer reviewed. It underwent a process where peers, other experts in vaccines reviewed our data, evaluated it for accuracy as well as our conclusions regarding our vaccine, that they were in line with what our data shows, and it's been, vetted by these reviewers, and what's published now in Science Translational Medicine is a fully peer-reviewed paper. So.
So what about you? Is it exciting for you?
Oh, extremely. Yeah. Relative to Deb, I've been in science for a very short amount of time, and so it's exciting to be able to work on something that potentially has the potential to go into humans and actually make a difference, as opposed to the work that I've been typically doing it's all proof of concept work, right? We establish proof of concept in animal models, and if the data is good and if there's a market and a need, then those products move forward into humans, and it's very rewarding to be able to work on something that will eventually be used in humans, hopefully.
Yeah, I think that we're undergoing a vaccine revolution. COVID-19 has brought this on. I've been in vaccine research for many, many years, and I've never seen anything like this. We are moving at much quicker pace than ever. So there's a revolution in that way in terms of how quickly we can produce vaccines. But at the end of the day, you're seeing a lot of new vaccine technologies being brought to the forefront. And once we've actually shut down this pandemic, what we're going to be left with our amazing set of new vaccine technologies that we can then use for future pandemics, for infections that we don't have vaccines yet for, like HIV or malaria, or even for cancer. These are all showing significant promise, and I think it's really just going to accelerate, the field in that regard.
Yeah. Yes, we can confirm that's.
Can you repeat that?
So yeah, the question is a clarification. Are human trials going to begin later this summer? And as of right now, the plan is to begin trials later this summer with HDT Bio.
And then how long would it take based on that once you begin trials? Do you have a sense of timing after that?
That's outside of my area of expertise. I think the plan is to move as quickly as possible, very kind of following in line with a lot of the other companies where they're concurrently starting phase 2 and phase 3 trials. But honestly, I don't have that information in front of me.
Yeah, traditionally vaccines went through phase 1, 2, and 3 clinical trials. You would always start the phase 2 after the phase 1 was finished. That resulted in about a 5 to 7 year timeline. As you're hearing, now vaccines are being accelerated through all of these steps within 18 months to 2 years. So we're going to be following a lot of the same sort of strategy where you overlap sort of the phase 1 to phase 2 and phase 3. And that introduces risks not in terms of safety of the vaccine, because they're still going to meet all those check marks in terms of safety, but it does pose a risk for those manufacturing the vaccine, because they're going to be producing a bunch of vaccine and put it in a warehouse anticipating those clinical trials will work. And if something comes up that says that they're not, or they're not safe, then they're just going to have to flush all that down. So
What's the earliest you think we can see this? Presuming all the clinical trials work, it's safe and effective, how soon can we see this?
this particular vaccine, I would say, if it follows the same trajectory as other clinical trials, which are being accelerated around 18 months or so, then we can see from 18 months from the summer, maybe a year from now. And, we can anticipate that as, we learn more about how quickly we can move vaccines forward, that we might, HDT, as they begin to move this vaccine forward, might benefit from the pipeline that has been opened up by other vaccines that have started before this one.
I think there are some people who may be confused because we are seeing headlines of vaccines. They started research a few months ago. Maybe they'll be ready by the end of this year. Mm-hmm. This one seems to take long.
Well, I think that what's confusing is that what they're looking at are vaccines that were already potentially in phase 1 trials for another indication, and so they could quickly pivot that one by just simply swapping out the sequence. What we've done is we've introduced an entirely new vaccine platform, which we think is going to provide superiority over some of the other existing ones, but that requires us to start in preclinical studies in animals, which is what we reported today. Yeah.
Can you elaborate on that?
Yeah, a lot of studies that you see coming out of new vaccines, it seems pretty exciting, but they're just reporting their data in mice, and mice are notorious for not really predicting, results in humans. So, with monkeys and non-human primates, they are our closest model to humans, and history has shown that doses and vaccines and vaccine immune responses that work well in monkeys translate very well to humans. So our study showing real good promise of this vaccine in a larger non-human primate model is very promising for its potential to work in humans.
Can you talk about how many people enroll in the clinical trial, and then again repeat the uniqueness of what we found in this study compared to others out there?
Yeah, usually a phase 1 trial is going to start with about 40 to 50 people. Generally, it's a dose escalation study that allows you to determine what is the optimal dose for immunogenicity without causing reactogenicity or safety issues. And so that's the first step. Phase 2 generally goes into hundreds, even thousands of individuals, and a lot of times that will include additional demographics like the elderly who may not respond as well to typical vaccines. The phase 1 is usually in young, very healthy people. And then the phase 3 trial is one that's done in tens of thousands of people, and it's going to include a placebo control to actually determine if the vaccine is effective. Does it actually reduce transmission in the individuals that receive the vaccine? So those are sort of the steps that we go through. To reiterate what are the key advantages of this particular vaccine, we think it's twofold. One is that it's a replicating RNA vaccine, and that could potentially allow us to immunize with much lower doses and fewer doses and achieve protective levels of immunity. The replicating feature of it also may allow those antibody responses to be more durable. We saw initial evidence of this as we reported in our paper because the antibody responses came up within 28 days and they continue to increase with time. And so, like I said, we don't know for sure how long those antibodies will persist, but theoretically a replicating RNA vaccine may provide more durable antibody than, say, a non-replicating one, which is what most the current clinical trials are using. The second piece is really the formulation, where we theorize that it may be more scalable because of the way that it can be stored and it can be produced with the RNA actually going on the outside of the particle instead of having to be formulated on the inside. That makes the process less complex, allowing the mixing to occur right at the bedside instead of having all that mixing having to occur during the manufacturing process, which increases the complexity and potentially could provide a bottleneck. So those are sort of the main features. Our RNA vaccine we've seen induces antibodies as well, T-cell responses. This is consistent with what other studies have, other groups who are developing RNA vaccines have shown that RNA vaccines are very well known for inducing both antibodies as well as T-cell responses.
Are there any other RNA vaccines that are commonly used today?
Yeah, so there are no licensed nucleic acid vaccines, but we need to include in that scope both DNA and RNA. And DNA vaccines, which is got started actually quicker than RNA vaccines because they're more stable, they have been actually licensed for veterinary use, and there are several DNA vaccines very far along in clinical testing for immunotherapy, for example, of cancers and the like. And so there's anticipated that probably a DNA vaccine might be licensed as well as an RNA vaccine for COVID-19.
It seems like this would be the first widespread use of a nucleic vaccine.
Yes, and that's the exciting part to me, having worked in nucleic acid vaccines for all my life, that this is actually what I say is the revolution, is that these technologies have been cooking for many years. We've been working on improving them, making them actually work better in humans, and now they're going to come of age here. We'll always remember the pre-COVID and post-COVID days, I think, for us vaccinologists, because it's just really accelerated the development of these technologies.
One last question.
How long has the primates those
How long have the non-human before dose and after dose?
Wanna take this?
What are we up to now?
We continue to monitor these animals, and the study began a little over 100 days ago. So we plan to monitor them continuously through at least August, and then the plan is to enter them into a challenge study to see if the durability of immune response is protective upon challenge.
Yeah, so at that point, we'd probably be about close to 6 months, 5 to 6 months after the vaccine dose was administered, which is consistent with the window of time that you'd want a vaccine to be effective. At least a 6-month durability and protective efficacy. So.
And just to clarify, challenge study means you'll expose them to COVID 6 months or so after they've been vaccinated.
Correct. Yeah.
See if the vaccine still works.
Exactly. Non-human primates are very similar to people, both in their response to vaccine and also their susceptibility to infections, including SARS-CoV-2. They exhibit the same kinds of symptoms that people do, and so they provide an excellent model for us to evaluate whether our vaccines are actually going to provide protection. As you know, we can't challenge people, and mice are not actually naturally susceptible to SARS-CoV-2, so the non-human primate model plays a very important role in developing COVID-19 vaccines.
Any other questions here? Any
Thank
other
you.
questions in the chat? All right, thank you, everybody.
Thank you.
you.
Thank you. It was a pleasure. Yeah.